Right, core concepts in class taskbook and go out with a bang. Essay, question 10. It's one suggested that proteins are produced by ribosomes, which translate messenger RNA molecule produced by transcription of a single gene. So as a statement itself, it's not too bad, is it? You know, proteins being made by ribosomes, just transcribing genes, so it's not too far away from the truth. With reference to your knowledge, ka-ching, an understanding of protein structure and synthesis. Explain why the statement above does not fully okay, so it's saying it is okay, in bold, fully describe how functional proteins are produced and secreted by eukaryotic cells. So there's quite a lot of stuff to talk about in that statement that you've got the knowledge for. So we're not going to write out the essay for you. We're just going to suggest some areas to talk about. The cash-in of your knowledge is knowledge of protein structure and synthesis. So I'm definitely going to talk about protein structure. So I'm gonna say the primary structure, amino acids, the condensation reactions and so on, to make the secondary structures. I'm going to talk about alpha helix and so on. I'm going to talk about tertiary structures, further bending and folding, bonds between different bonds like ionic bonding, disulfide bonding between R groups, and I'm going to talk about the quaternary structures, more than one polypeptide chain. I'm going to think about other prosthetic groups like heme groups and things like that in hemoglobin, some examples that you might know. So that's my knowledge of protein structure. And then synthesis. So protein synthesis. So I'm going to go back to transcription, translation. It mentions translation, but so that's our knowledge. And then it says functional proteins. So it tells you that for a reason. So it's not just this primary structure that it's talking about, being made by ribosomes. It's adding of these other groups and changes to make fully functioning enzymes and things like that. And then we've got produced and secreted by eukaryotic cells. So we're going to have to, secretion. So I'm going to involve other organelles now. So I'm going to think about endoplasmic reticulum. I'm going to think about Golgis, vesicles, exocytosis. So that's my knowledge that I'm really gonna use here. So problems with this statement now become slightly more apparent. The fact that we've got quaternary structures, so more than one polypeptide chain, so we're going to need more than one gene to do this. And so we have one gene for one polypeptide, and so if there's more than one polypeptide there, like for example hemoglobin has got two different types of chains, I'm going to need at least two genes for that. So that's a problem. So we need more than one gene for those. And we've got functional groups like heme and hemoglobin and things like that, or some other proteins might have carbohydrate groups or other things attached to them. So other groups, and that's certainly not covered by our statement with the ribosomes. That's going to be perhaps in our Golgi. Possibly we tell you the definition of your Golgi, is modifying proteins, packaging proteins. So we could mention that there, and go through our list of organelles and what they're doing to produce and secrete out of a eukaryotic cell. So those are sort of the three areas I would talk about, but it's a good opportunity to put a lot of your own knowledge about protein structure and synthesis and secretion in this essay. Good luck.